Hello lovelies, in this video Dr. Upwards is going to be going through continuous and discontinuous variation for your A-level biology. So lots and lots of examples in here to really help you understand and be able to approach these questions in an exam. Now, if there's anything you're confused about, you can watch the video again, and if you want to test to see whether you've thoroughly understood this, whether you've understood it enough to be able to approach this in the exam, then over on the website, Dr. Edwards has given you a special set of questions just to go with this video.
The genetics of continuous and discontinuous variation. So we've talked about and mentioned both continuous and discontinuous variation before when we looked at different types of variation. But what we need to do now is take what we've looked at and appreciate now with the alleles and the different gene loci that we've looked at in this last bit of the topic to kind of think about how we can explain the genetic basis for both types of variation. So discontinuous variation produces discrete phenotypes. So there are two or more non-overlapping categories. So for example, you can either have A, B, O, or AB blood type. There are no intermediates there. Or you can have attached or non-attached earlobes. These are the two examples of like discrete categories that you have to be in for discontinuous variation that are caused by specific genes. Usually, discontinuous variation is controlled by monogenic traits. So this means it's determined by the allele of a single gene locus. So if you look at my graph here, which we've looked at before, we've got three categories. So this is going to be the three phenotypes normally. And so you've got homozygous dominant, you've got homozygous recessive, and then you've got heterozygous in the middle. So these, if it's one gene, single gene locus with maybe two alleles in this case, then these are the three options or the three categories you can be in genetically. And then that will determine your phenotype, which will be, again, a few categories. We know now that sometimes the alleles of two genes can interact and affect a single characteristic though. So we've looked at this as an example with epistasis. So this is where the genes at different loci can interact in order to bring about a phenotype because one could mask the other. And also there could be different alleles at a single locus that will create the different groups, okay? So obviously we've got A, B, O, and AB and their interactions. So there's multiple alleles there, but it's still a single gene locus. But the different alleles you can have and the different combination of those alleles you can have will cause changes to the phenotype. Okay, so let's contrast that continuous variation. Continuous variation between organisms is often made up by quantitative traits that vary by very small amounts. So that means they're measurable traits as opposed to just being in a category or not. So the small variation between individuals in the population means that we get this smooth, graduated curve if we plotted it as a curve rather than a bar chart in the range of phenotypes across the population. And that's how we get that kind of bell curve that we see. So we think about things that we can measure quantitatively and that can have very small increments of change. So mass, height, the length of a corn cob or a seed pod in a plant. All of these things are examples of continuous variation. The reason for this small incremental changes is that these traits are polygenic. That means there's many genes involved in determining these characteristics, not just a single one. The different alleles of each gene only contribute a small amount to the characteristic, but it adds up, so there's an additive effect. So that means that there's variation in all of the genes that contribute to this characteristic between the different alleles you can have for each gene and the amount that they contribute to the actual characteristic. And then you add them all together, so everyone's going to have a different sum, if you like, of the alleles and the effects of those alleles on their phenotype. So you can call this quantitative variation because the more gene loci that contribute to the characteristic, the greater the range in phenotypes there's going to be for that characteristic. So the more genes involved, or the more alleles of each gene and the more genes involved, the greater the range of phenotypes we're going to see for a characteristic. So the greater the range, the more bars you would see on this kind of bell curve shape, the more genes there are, the more phenotypes there are, which would mean the more bars there are. The role of the environment. So we know that the environment plays a role in gene expression and also phenotype. The environment is going to have a greater effect on the expression of polygenic or continuous characteristics. This is because the added effect of environmental pressures can increase the range of variation in a characteristic further, e.g. height. So we know that nutrition or the availability of nutrients that you have during growth periods, whether you're a human, for example, or if you're a plant, that's going to determine how well you can grow in terms of height. And it is a way of affecting that genetic predisposition for how tall you could grow. So in humans, there's over 400 genes that have been linked to height, which is why we have such a massive range of variation in phenotypes of height. And your predisposition, genetic predisposition, is going to be changed and determined by the nutrition that you have during childhood. And this is just one way that the environment is interacting with genes in order to play a role in what the eventual phenotype will look like, and therefore produce even more variation than there would be just from the 400 genes, creating that additive effects of differentiation over the population. And this is the kind of one way we can describe how the environment plays a role. And it only really has this opportunity to play a role in the polygenic characteristics because there's so much variation already. And your kind of environmental condition, unless it's something really, really, really specific, wouldn't be able to change what category you're in if there's only two categories, unless there's something involved like a mutation, for example. But it's unlikely to have as much of an impact because there's not much it could change. So I've got another example here with the height thing with plants. In terms of that, if you think about the fact that these two plants could be genetically identical clones of each other, but we've put them in different soil nutrient levels, so one's got low nutrients and one's got high nutrients, the one with high nutrients is obviously going to be able to grow taller. And that's nothing to do with its genes, it's going to be to do with the environment that it's been placed in. And so we need to be able to understand that these interactions can occur and just increase the variation further on top of the already genetic variation there is in the population.
Ouch! This is why in some videos I explain scratches.